Thank you very much, Dean, for that introduction. It is indeed an honor and a pleasure for me to address you this afternoon on some of the work that we've been doing. And really, the question is good science going nowhere? And I strongly believe that in a country such as ours, where at all possible, and in some instances it's certainly not possible, and I accept that, but where at all possible, we really have to try and implement the outcomes of our research to address the many challenges that this country has, whether it be environmental challenges or social challenges. And so it is that I put together this talk this afternoon and really came about due to a news coverage on this article here on what is seen as a new invasion of an aquatic weed in South Africa on the Vaal River, and the response that you can see in the photograph below of a herbicide application. Now, South Africa is not new to aquatic invasion, and it's not new to aquatic invasion of the plant nature. And we have many sites around our country which are invaded by aquatic weeds. This is a particularly favorite site of mine up near Stanger, completely covered with water hyacinth. And going back to the 1960s, we've been fighting against biological invasion in our freshwater systems. And this brought about what became known as the Big Bad Five, the red water fern, Salvinia molesta, Myriophyllum aquaticum, and Pistius radiotes, with the main villain of the piece, water hyacinth. Now, since the 1970s, we've been trying to address these weeds through biological control. And we've got a range of different insects that we've introduced on a whole lot of them. I'm not going to go into the science of biological control, but suffice it to say that these agents are thoroughly tested before they are released into the environment. And on water hyacinth, we have the first two most important insects, the Neocotina weevils. We've also introduced a mite pathogen, a moth, a couple of mirid species. And this little insect here is the most recent introduction, Megamelis, one that I'll be talking about a little bit later. And with this biological control agents available to us, we've had some success. This is a site close to Rhodes University near New Year's Dam in Alisdale. And just with the introduction of Neocotina eichhornii, we've been able to move the site to this. And you're going to see a lot of before and after photographs in my presentation this afternoon. And what I'm going to hopefully convince you during all of these wonderful before and after photographs, that it is not the wind, it is not winter, it is not herbicide, it is not mechanical control, it is not some other intervention that has simply removed the weeds from this site, that I'm going to show you some data that supports these wonderful before and after photographs. So we've had some success with the biological control of water hyacinth. Many of you will be saying, yes, but what's happening at Hartbeespoort Dam? And I'll refer to that in a moment. What's happening at Roodeplaat Dam? What's happening at many other sites around the country? One of the messages I really want to get across here this afternoon is that it's not one size fits all. All of the sites differ. They differ in seasonality, they differ in temperature, they're different in nutrient status, and this then has an effect on how effective our biological control agents may or may not be. We've had good control against Salvinia, giant Salvinia, with the introduction of Certabagus, reducing the sites to looking like that. Parrot's feather is the one weed that we work on that's not floating. It's attached to the substrate. Also, introduction of a beetle in 1994, and these sites get cleared up really quite easily. And there's some trends that I'm going to pull out later as to why we get this level of success. One of my favorites, red water fern, or Azolla folliculoides, introduced in the 1940s, and this tiny little weevil, Stenopalmus, which was released in 1997, thanks to some excellent support from the Water Research Commission, and was able to clear this weed not only at this particular site here, but across the country, as we will see later. So back to the villain of the piece, and the reason why I decided to give the title of my talk a fairly provocative title. This is Pistia stratiotes, water lettuce, also an invader from South America. It's been here for a long time. It's not a new invasion. The invasion on the Vaal River is fairly recent, but it has been present throughout the country, probably going back to the 1960s. We've got a really good little beetle, Neohydronus affinis, which was released in 1985. And once again, lovely before and after photographs that I can show you. And in fact, this biological control program became one of our flagship programs. This was work done by Karina Cilliers back in the 1980s when Neohydronymus was released at Sunset Dam in Kruger National Park. And here you can see I've cobbled together a couple of slides of hers, once again, a before and after, showing very clearly that this insect can be an effective tool. So with this in mind, back to this slide, it came as great concern to me that one of our water bodies was considering spraying water lettuce on the Vaal River. In fact, today, some spraying of water lettuce has occurred. So what this does was made me to, I guess, sit down and think a little bit and say, well, actually, is the biological control of water weeds as effective as we think? Is there something else going on here? Maybe it works too slowly. Maybe it's not rapid enough for the people utilizing the water body to employ biological control over herbicide application. Is it cost effective? Maybe it's cheaper to use herbicides. Are we implementing it correctly? Have we taken the science from the lab bench into the field? Are we communicating it correctly? Is it just that people don't know that we are about biological control, that people don't understand what biological control is and how effective it may be? And so it really comes down to the question of why would a water authority choose to use herbicide over biological control, given the series of slides that I've just shown you, and taking you on a journey to say that biological control really works. And I guess at the end of the day, some 20 odd years after starting to work on these programs, maybe it's time for reflection. And so how effective does it work? And one of the problems that we have with biological control is that it's kind of it's an all or nothing. Once the insect is out there, the insect is out there. And so it's very hard to conduct what we would call manipulative experiments. And so we're left very much with a before and after scenario, which many of you will know is really quite difficult to first analyze statistically. And secondly, there's the old adage of correlation versus causation. So we go out to these sites where we've released these biological control agents. This happens to be Hartbeespoort Dam in the slide here. And we collect a whole lot of environmental data, biodiversity data, plant population data, biocontrol agent data, and economic data, and so on. And the big thing here is data, data, data. But the problem is, what do we do with it? And how do we analyze it? As I said, much of the data that we get in is very much before and after designs, and it is open to criticism, in particular around, was the biocontrol agent actually the causative agent? And so what we need to do is do long-term monitoring, and that's a little bit of the data that I'm going to show you now. We have done some insecticide exclusion experiments. I'm not a fan of insecticide exclusion experiments. I think there are all sorts of other factors that are at play there. And this, if you can see, if the slide is clear enough at the bottom, on the left-hand side is a mat of water hyacinth that has had an insecticide applied to it to remove the biological control agents. And on the right-hand side is a mat of water hyacinth that It hasn't had an insecticide on, and the biocontrol agents have built up. And so we can collect these sorts of data. We can show that there's no agents on the left. The plants are a lot taller. The plants are expanding. On the right-hand side, there's lots and lots of biocontrol agents. The plants are a lot smaller, not flowering, and so on. So we can do insecticide exclusion experiments. But really, the important thing is long-term monitoring data. And so we set out some time ago, 2008, and we set out to undertake long-term monitoring. For Azolla, we've been doing it for longer. We started it right when we released the first agent in 1997, and we conduct countrywide surveys. We go to as many of the sites as we can around the country to collect data. The data are very simple. It's really measurement data, in the hope that we can show some long-term trends. And we've done that. And I've just put, I really hate it when people do this, but I didn't know how else to do it. So I put a whole series of publications up here. And what this is supposed to show is that we've collected the data, we've analyzed the data, and we've shown, hopefully, the impact of the biological control agents on the respective weeds. And if anybody has far too much time on their hands and wants to have a look at these, they're welcome to get hold of me afterwards, and I can send you some of them. But I just want to take some of these examples, just go through some of these examples of these long-term data. Now, this isn't a huge data set. It's only about 7 or 8 years, and it's of Azolla folliculoides at 91 sites around South Africa. And what we can see here is what we've measured is the area of infestation, and the starting point is 100% cover by Azolla folliculoides. This is the number of agents that we've released. This is the number of sites that we've controlled, and 91 of the sites, or 81% of the sites, we have controlled Azolla. Now, what is control? And we'll talk about that in a moment. But here, what we've looked at is the number of hectares cleared. And what you can see is that of the 215 hectares of Azolla that we started out with, we've managed to clear over 203 of those hectares. And so in a lot of ways, when we started these biocontrol programs, it is, what is your measure of success? How do you measure success? Is it, in this case, the surface area of the impoundment, the river, the dam cleared, or is it some other measure? We would say, even as the agent establishes, that's some measure of success, but it's really how we measure the success that's important, and I'll get onto this in a moment. And one of the really strange things about the Azolla example is the amount of time that it took, that we were able to clear these sites in a very, very short space of time. And in many ways, the Azolla example has, I guess, set us up for failure, in that it was very, very effective in a very, very short period of time. So what's happened is we've now, taking this data set a little bit further, we've eradicated Azolla folliculoides from 105 sites around South Africa. There's a reason for that, and that is that this is Azolla is a fern, and in many instances it doesn't produce sporocarps and doesn't produce spores. Where these plants don't produce spores, we are able to eradicate Azolla from that site. It is not a classical biocontrol example, as it were, where we would expect a residual population of the plant to remain, and then as the insect population dies out, so the plant increases again in that typical density-dependent relationship. That is not the case here. And the case here is that where Azolla doesn't produce spores, the weevil, in fact, can eradicate it. As I said, the weevil disperses well. There's no need for us to mass rear it. And in fact, we are busy with the Department of Environmental Affairs, Forestry and Fishery, as we speak, DFFE, sorry, in trying to get Azolla folliculoides delisted as an invasive alien plant in South Africa. It is no longer invasive in South Africa because of the impact of this weevil. Salvinia molesta is another classical biological control program, not just in South Africa, but all over the world. And what this graph here is showing you where the red arrow is here is when we began our countrywide surveys. And here we can see that we had, this is the number of sites of Azolla in the black dots. And you can see this huge jump between before and after when we started. This is because a lot of this data here on the left-hand side of the graph was reliant on SAPIA data, the Southern African Invader Plant Atlas data. And when we started doing post-release evaluation, obviously the number of sites that we looked at increased because we were actively looking for it. But what this graph shows is that over time, the number of sites have actually increased. And this is because we are looking for it, also because Salvinia is probably dispersing to these sites on waterfowl and on boats and so on. But what is important here is that the percentage cover at these sites has significantly decreased over time. So although Salvinia still remains at some of these sites, and we're getting more and more sites, the percentage cover at the sites where we've implemented biological control has massively decreased. So the number of sites increased over time. Sites at these cover at these sites has significantly decreased, and we've in fact eradicated this weed at 37 sites. Once again, this was only possible because Salvinia molesta is a sterile polyploid. It doesn't reproduce sexually, it only reproduces asexually. And so there's no residual spores left in the system once the insect has controlled it. What of Pontidaria? What of water hyacinth? Many of you might have seen some of these data shown by Julie Coetzee. This is her work and the work that she's doing on Hartbeespoort Dam. And what's happened on Hartbeespoort Dam is a really interesting example whereby up until 2017, biological control was very, very ineffective. In fact, completely ineffective on Hartbeespoort Dam. There are a number of reasons for this. The first was the fact that the dam is highly polluted and the plants, the water hyacinth plants, grow excessively due to that pollution. Secondly, it's in the Highveld, it has cold winters, and the cold winters really knock the insects back. But thirdly, and most importantly, is that up until 2017, the dam was under very effective herbicide application. And in this instance, the herbicide application was contrary or antagonistic towards the biological control. Since 2017, what's happened is we have introduced a new biological control agent, this little plant hopper down the bottom here called Megamelis scutularis. We've not only introduced it, we've mass reared it in huge numbers, as you'll see in a moment. And what we have been able to show over time is the reduction, seasonal reduction in water hyacinth. Water hyacinth still becomes problematic early in the spring and into the summer as we try and introduce as many of this insect as we can. And it's by late summer that we get really, really effective control. And I would refer you to Julie's work on this. And we can see this classical curve whereby this is the water hyacinth line here. And we can see that January through February to the beginning of winter, the water hyacinth populations really, really grow. In this instance, in 2023, we have the introduction and the numbers of Megamelis, the number of Megamelis per meter squared, and we get a resultant reduction in water hyacinth. There's no doubt that this is also compounded, mortality of the plant is compounded by the onset of winter as well. And once again, in the second season, we can see much higher populations of the plant hopper, and we don't reach the same peak. And this is a short period of time. We do have more information going back, and we're still collecting information, but we're hoping that this reduction in the peak from 2023 to 2024 will continue. We're not finding flowering of the plant. We're hoping there's less seed in the system, but this is one plant that does recruit from seedlings. There are lots of conspiracy theories out there. There are discussions that, in fact, it's not biological control, that it is, in fact, herbicide application, mechanical control, or some other control. But this slide, taken in 2020, shows where we have biological control of the water hyacinth. Everything that you can see in that slide that is brown is dying water hyacinth, and the green is in fact another plant. And were it herbicide application, we would anticipate that the other plants would have died as well. So there's a lot of politics that go on with this, and I'll refer to that right at the end of my talk. So once again, what of water lettuce, the plant that is being sprayed on the Vaal? Well, maybe it's that the Vaal is too polluted and biological control can't work. Well, this is Cape Recife Sewage Works just outside Gqeberha, formerly Port Elizabeth. And what I really want you to do is just see those two palm trees there. Keep those palm trees in mind as I go through this. Highly, highly polluted, lots and lots of water lettuce plants. The standard measure is a flip-flop. That's a 9.5 flip-flop. So you can see that the plants are really, really large. We haven't got round to publishing our work in flip-flops yet, but maybe that's as a unit of measure, but maybe that's something to come. We introduced this tiny little weevil, Neohydronymus affinis, in 2002. We started to see damage, and these are the shot holes caused by the adults and the larvae causing mining. And this is some of the work that we did where we looked at percentage water hyacinth cover. So we started measuring in April 2003. Remember, the insect was released in August 2002. So after about 7 months, we've still got 100% cover. But over time, we have a reduction in that. And this is all the way through the summer months. And here what's happening is that as it's getting cooler, into winter, so the insects don't do as well, and the plant comes back. But then once we get into summer again, the insects start to catch up with it. So this is percentage cover, and that's important here. We also looked at the mean mass of the plant, and we can see early on that we've got quite large plants, and as the insects really take hold, we get less and less of the lighter and lighter plants. What's really important here, because like water hyacinth, water lettuce produces fruit and it produces seed, is that once the insects take effect, we've had absolutely zero fruit production and obviously absolutely zero seed production. So the hope here is that in time, we reduce the seed bank in the sediments. They will continue to germinate. The insects will get on top of them, and hopefully over time we'll get effective control. There are those two palm trees I asked you to talk about, to remember. I took the original photograph from that bank of the far side here, and you can see that here we've still got some water lettuce recruiting from seedlings, but even in a highly polluted, non-tropical site, such as waterworks, sewage works in Port Elizabeth, we can get effective control. So we can get effective control here. What's happened around the world, what's happened internationally. There's a really good catalog, which is kept up to date by Winston. In fact, this is the published catalog, but it is online. And these agents have been released throughout the world. And we've recently re-looked at how we assess biological control, and how we assess the success of it. And we really believe here in South Africa now that with parrot's feather, with Azolla folliculoides, with Salvinia, and with Pistia, not only here but in the rest of the world, we've in fact reversed invasion. We've not slowed invasion. We've in fact reversed it, that at many of the sites, although you saw there was a cumulative increase in the Salvinia sites, that in fact that is slowing now, and we predict over time that there will be less and less sites. So not just us in South Africa, in fact 39 countries around the world are using biological control of water hyacinth. This is an example from Senegal. This is water lettuce control in Senegal, mean number of plants per meter squared. And here we can see the number of weevils, the number of plants per meter squared, sorry, and the percentage of those, or the number of weevil damaged plants per meter squared. We can see an increase in the number of weevil damaged plants per meter squared, and we get this massive reduction once again in the infestation of water lettuce. This is some work done by Oceana Diop in Senegal, and but published with us. So we think it does work, but is that enough? Is it enough that we control an aquatic plant? What about ecosystem restoration? What happens once we control an invasive alien aquatic plant? And we've only recently started looking at this, and we've started looking at how do we get a response in the aquatic ecosystem? And a couple of systems that we looked at, and it's by no means comprehensive yet. A number of systems that we've looked at, we've shown that no, if you remove the macrophyte, the floating macrophyte biomass, you get a response by the benthic invertebrates and by the ecosystem. And so this gives us confidence that in some of our systems, we might actually be able to restore them to something a little bit what they were originally. Obviously, there are disturbance factors. There are disturbance factors of pollution, increase in nitrates and phosphates in our aquatic ecosystems, and other disturbances as well, including introduction of new invasive alien plants which respond to that. We've also looked at the economic impact of these. This was some old work that we did on Azolla, and we showed the impact of a mat of Azolla folliculoides, what impact that had on everything from the prices of houses on golf course estates, all the way through to blocked pumps, blocked pipes, reduction in irrigation capacity, and drowning of livestock walking into this. And what Andrew McConaughey showed very clearly for the year 2000, so it's old data, that the benefit to cost ratio was calculated at 2.5 to 1. So for every rand that the Water Research Commission put into this, the country accrued 2.5 rand back. But as we put more effort into it and left the insect to carry on and do its thing, this benefit to cost ratio increased over time. We've also recently done some work. This was a master's student in economics, in resource economics at Rhodes University. And what Mary did, and it's deeply ironic that we published this a couple of years back when we hadn't sprayed Pistia for probably 20 years. And today we are spraying Pistia. But anyway, what Mary looked at, she said, okay, let's have a look at 4 of these weeds where the biocontrol agents are really effective, that being Azolla, Pistia, Myriophyllum, and Salvinia. In the absence of biological control, one or other government departments would have intervened and they would have used herbicide to control these weeds. Let's look over a 20-year period and see what the herbicide saving would have been to get these weeds under control to the same point that they would have been had we sprayed herbicides. And so she ran a whole series of different scenarios of the different weeds here, whether we looked at, and we offset that against biological control. So we looked at biological control as a cost as well. Whether we sprayed by boat from Bucky or a knapsack, many of these weeds are in small infestations, in small impoundments, and thus not requiring aerial application. And she said, right, so and she discounted it. She used a couple of discount rates. I think she used 5%, 8%, 10% discount rates. And she looked at it, and we looked at the most conservative number. And what Mary was able to show was that over the last 20 years, South Africa has saved over R1 billion in herbicide application on these 4 specific aquatic weeds. And I always put this in just to help our international visitors. So we've saved over R1 billion. That is a lot of money, but it's not that It's if we sat down now and bought R1 billion worth of herbicide, I don't know how much that is. I really don't know how much a liter of herbicide costs, but Mary obviously did. That would be a huge amount of herbicide that we have not poured into our aquatic ecosystems. We didn't look at the biodiversity benefits of this. We purely looked at the cost of herbicide it would have taken to control those weeds. So we know it does work. We know that there are benefits to doing it. Maybe it takes too long. To clear the azolla sites took under a year. To clear myriaphyllum sites take 1 to 2 years. To clear salvinia sites takes 1 to 2 years. This is the reason for this, as I've already explained, is that none of these three produce seeds or spores. And so they reproduce purely asexually. And as a result of that, we are able to get localized eradication of these weeds at those sites. Pistius takes a little bit longer, 4 to 5 years, and Pontidaria, also the sites can be cleared annually, but they do return from seeds. And the average time we take to clear, we actually don't know yet. There is discussion that seeds of water hyacinth are viable for up to 20 years in the sediment. And we've shown that there are high numbers of seeds, up to 2,500 seeds per meter squared at some of our sites. And so we're in this, if we're in it with biological control, we're in it for the long haul. So some of these weeds recover from seeds, and some of the agents as well are poor dispersers. So that then goes to implementation. Are we maybe not implementing biological control agents correctly? We've taken an approach through the Center for Biological Control that with many of these floating aquatic weeds, we are moving away from a classical biological control approach, where you put the agents out and you allow the agents to build up on their own over time. We have taken on an augmentative approach, and this is important for us in South Africa because we have these highly eutrophic waters. We have cold winters, which result in mortality of the insects. We have floods, we have droughts, we have interference from herbicide application, and in many instances, we have these seed banks. And so what we do is we mass rear these insects and we release them seasonally. So we release them throughout the year, but we release them wherever they're needed. Obviously, for this stakeholder involvement is very important. Excuse me. But the one thing it does is it offers training and employment. And we've been able to employ a lot of people through the mass rearing of biological control agents. Not only that, is we've looked at this critically, and the Center for Biological Control has been very fortunate to receive money from DFFE for many years, initially through the Working for Water program. And one of the aspirations of the Working for Water program was to try and see how they could give meaningful employment to people living with and working with disabilities. And this is something we've been able to do. This is a picture from our Vinek research facility where these guys mass rear insects. And the people that you can see in the picture, besides Lyle and Samela, these are all people living and working with disabilities. And we've been able to train these guys to help us rear biological control agents. One of the first programs I worked on with this was way back, probably 20 years ago now, at a place called Warrenvale Hoërskool up in the Northern Cape at Warrenton. And what we did there was we took an old cricket net, we put a whole lot of paddling pools in it and a whole lot of water hyacinth, and we introduced a whole lot of biological control agents. And that is not somebody littering there. That is in fact a dispenser for fertilizer. And we introduced the insects and we mass reared the insects and we trained everybody. And this school is right on the banks of the Vaal River, and it's right on the banks of Warrenton Weir. And over time, this school released thousands, if not hundreds of thousands of weevils into this site, and were able to clear the site effectively. We've got a large-scale mass rearing program, as I say, working with people with disabilities, creating meaningful jobs for people with disabilities. We've also expanded this, and this is the example of Megamelis on Hartbeespoort Dam, where we've not only mass reared insects through the Centre for Biological Control at Rhodes University, but we've also set up mass rearing facilities around the dam. Not just the Hartbeespoort Dam, we're doing this throughout the country. And this does a couple of things. A, it gets a lot of insects into the field. B, it introduces people to the biology of the weed they happen to have on their dam and the biology of the insect. And C, it really gives people a sense of ownership that they are doing something for their own environment. And this is maybe Spider-Man can help us out too, although I'm not sure Spider-Man and rearing insects is a good idea. But this has been a very, very effective program that we've been able to roll out around the dam. And we've been able to then introduce a whole lot of insects into the system. So if we look, this is water hyacinth cover on Hartbeespoort Dam. Here's the percentage cover from February 2023 all the way through. And we can see there's a high peak here, up to 60% cover. And the water hyacinth does decrease during winter, not only because it's had a lot of herbivory on it late in the summer, but the plants start to take off again in October. And the problem with this is that our insects are slow. If we didn't mass rear insects, our insects get hammered by the low winter temperatures, and our insects only start catching up with the plants probably 2 months later. And so we end up with these peaks that we have to try and control. And so what we've been able to do is we've been able to, these are our average abundance of insects from our surveys is in blue, and the number of releases from our stations around Hartbeespoort is in yellow. And the whole idea here is that we rear insects under warmer conditions in tunnels and release them as early on in the spring as we can, and releasing here up to 140,000, 130,000 of these insects. And the hope here is that we don't then get these high peaks and that the peaks are somewhat attenuated. So we know it works. We know that it can have impact. We know that we can do the implementation. Maybe we just haven't communicated correctly. And I'm not sure that's the case. I mean, anybody here within the audience from the Royal Society would no doubt have, if you opened News 24, the Daily Maverick, any number of those fine mouthpieces of news, you would have seen even the cormorant. You would have seen the work that we're doing. And Professor Coetzee, who's had a very long and not very long, but a very well-published career, said her highlight, no doubt, was being on carte blanche. No matter what you publish or where you publish it, you haven't lived as a scientist in South Africa. You haven't achieved as a scientist unless you're on carte blanche. And so, and even today, as we speak, there were 2 inserts on SABC News around the mass rearing of cochineal for cactus control down in Kariga, formerly Uitenhage. And so I don't think we've done communication badly. Even the Mail and Guardian, the Times of London, these articles have been there. We have every social media platform that you can imagine. These articles are on. So I don't think we've done that communication badly. We've also done a lot of communication in terms of grassroots level. So we've got a young academic lecture series we run. We're very well represented formally at SciFest, National Science Week. We've got a high schools internship program, a science program. We bring scholars learners in to help us mass rear. So we've done a lot of that at grassroots level. We run a biological control short course, and between 2005 and 2020, we've put nearly 700 people through the short course. This is people from government agencies such as DFFE, Department of Agriculture, from our conservancies, whether it be sandparks, or the provincial ones. So we've trained a lot of people in biocontrol, talking mainly about how biological control is a safe and effective tool. So I don't think we've done that badly, but we still get this. So why are we at this point? And in a lot of ways, this is in complete opposition to what is happening in the rest of the world. The rest of the world is being driven into biological control, whether it be biological control of pests or biological control of invasive alien plant species. And this curve at the bottom shows synthetic pesticides over time, the predictions over time are that synthetic pesticides are going to come down and the utilization of biopesticides is going to increase. There are more and more biopesticide products in the USA and in the European Union, and also the European Green Deal, which seems to be shelved for a while, but pushing to say, can we halve the amount of pesticides in the world by originally 2030, but I believe that has been pushed back now. So what is driving this? In a lot of ways, this is being driven by commerce rather than the environment, that the consumers are increasingly discerning about the food that they eat. So we've got, on the one hand, we've got something we think really works. On the other hand, we've got the world saying we need more nature-based solutions, green solutions, and yet we're also spraying. Maybe it's history repeating itself. This is a mass rearing facility in Lusaka. This is one on the Shiri River in Malawi. And this is in the 1990s, as you can see from the poor quality slides. And the reason for this, the reason why we got involved in biological control of water hyacinth here is because biological control of water hyacinth had no future in South Africa in the 1990s. It was largely a pesticide regime. But there's sustainability here. There is, and this is Lake Victoria, this is Kisumu, and this is in 1995, massive amounts of water hyacinth. We rolled out a biocontrol program there between 1995 and 2000, and going back in 2017, the boat hasn't changed, it probably hasn't moved, but the water hyacinth has become under some effective control. We're under no doubt that this is driving our systems, and we've studied that. We've looked at the role of eutrophication. We know that eutrophication can limit success of classical biocontrol, but we're not in classical biocontrol anymore. We're using more augmentative biological control now. So we know that we can overcome that. The deep irony for me, this is our disabled program in Makanda. Here are 3 people with disabilities, previously never employed, producing insects, neohydronymus for water lettuce, that are being released in Morocco, the first ever release of a biological control agent for an invasive alien plant in Morocco, and recently controlling a site very effectively in the Masai Mara in Kenya. So I finish with this statement by a really good colleague of mine, who said that water hyacinth, waterweeds is my parentheses, is invariably a political problem and not an environmental problem. And really, I guess it comes back to this classical curve. And what we have here is a high population of the weed. We release the biocontrol agent. You've all seen this. We want to try and reduce the plant population below some form of damage threshold. And all I can think is that authorities really do want to push the white line through the x-axis and eradicate where they can. Maybe the timeline is too long. I don't think that's the case. Biocontrol is not the panacea. We do need other interventions to control invasive alien aquatic plants in South Africa, such as nutrient control, such as hydrological control. It does take time, but I think we've shown that it doesn't take. Some of these sites, like Hartbeespoort Dam, has had water hyacinth on it since the 1960s. And now we want to try and we're concerned that biocontrol can't remove it in a year or 2 years. Maybe we just need to be a bit more patient. We know it doesn't eradicate. It can form part of an integrated approach. We're not far down the line with that, and we need to look into that better, but it is sustainable. With that, I'd just like to thank some funders, and I'm very happy to answer any questions that you might have.
My pleasure to thank Professor Hill for his talk. If you'd like to ask questions, please put your hands up or put them in the chat, and I'll address them when I can see them. Let me start off. That data that you were presenting on the control of both hyacinth and in the sewage plant, I presume that is one application of the insects, and you just have one application there, not an application each season.
No. Not at all, Dion. The water hyacinth on hardebeestpoort, we are releasing biological control agents all the time. The example of the Cape RSSAf sewage works, we have had to make one or two other applications, and as I said, it's because Pistia water lettuce has seeds, so it does come back. And we just like to increase the population size if we can. But with the other three plants, the Azolla, the Myrophyllum, and the Salvinia, a single application of agents is sufficient.
And the insects themselves, they are so they struggle so much during winter that you have to reapply. They just don't grow fast enough, and that's the reason that you're reapplying early in the season so that you can get their numbers up as the plants start to grow as well. I guess.
Yeah, absolutely, Dean, absolutely. And we've done a lot of thermal physiological work on this. And, you know, all of these insects, although they've been here a long time, originate in the tropics. And we've actually had a look at not just on water weeds, but on many other weeds, trying to use tropical insects in higher latitudes. And one of the ways to overcome that is by moving away from a classical approach to a more augmentative approach, with the long-term hope that the pressure on the weed during the summer season will reduce the amount of plant vigor, will reduce the amount of, in this case, sexual reproduction.
And the insects that you're introducing, as you were saying just now, are those that are found in these environments anyway, so you're not introducing something that is going to cause other problems, I presume. Yeah. There's a question in the chat. Let me just call it up here. It's from Jasper Knight. Have you examined any wider ecological consequences of the presence of these biological control agents in these lake systems?
So, Jasper, yes, that's a very, very good question, and there's a couple of answers to that question. So first and foremost, one of the things that we test, obviously we test these biological control agents to ensure that they're host specific. One of the things that we don't publish is that probably around about 40% of the biocontrol agents that we've ever introduced into quarantine into South Africa, we have rejected because we haven't been able to show that they are sufficiently specific, and thus we haven't released them. One of the knock-on effects that we try and look at is the role that these insects play, maybe in the movement of pathogens. And one of the things we have shown, and in particular in the case of Azolla, is that it's not just the insect feeding, but the insect feeding opens the plant up to invasion by pathogens. And in fact, the pathogens is probably doing as good a job as the insect feeding. We have had a look at host range extension by indigenous parasitoid wasps onto biocontrol agents as well. We have shown that this is the case, but the parasitism levels seem to be very, very low in comparison to what their native hosts would be. So we do need to obviously look at that. We haven't looked at predation, so whether or not these massive increase in biological of insect food is having an effect, knock-on effect on the predator guilds, and this is something we probably will need to look at. But what we are looking at more is how these lake systems respond, the knock-on effects of these. So we've got a project at the moment. There was a big discussion around Hardebeestpoort Dam that the dropout of the mat of water hyacinth is likely to form a sludge, or I think the term was jelly layer on the bottom of the lake. We're showing that to not really be the case. There will be some allochthonous input. There will be some nutrient releases from the plants as they die. But our feeling on this, and once again we're studying it, but we haven't got the answer, is that the release is going to be a lot less than if you apply a herbicide. There's also been some discussion around application of sublethal doses of herbicides. And we've done some research on that, where we can reduce the concentration of herbicides such that we stop plant growth and allow the biocontrol agents to increase. This relies on the assumption that the herbicides and the insects are compatible, and that we're not going to cause mortality to the insects. We're still some way away from understanding that as well. So yes, there are obviously, we can't just look at this in isolation. We do need to look at it from an ecosystem perspective. These studies are very difficult, but it is something that we try and do where possible.
Thank you. I'm curious why, with the number of plants that are invasive aquatic species, or the plant species that thrive in different areas, is there reasons why some plants just seem to do particularly well in different parts of the country and others elsewhere? The plant distribution, is there reasons why there's more hyacinth in one part than say Salvinia in one place and Pantidaria elsewhere,
Yeah,
for example?
not really. I think some of it's a legacy of how it got there, but also probably a result of management strategies. For example, we get a lot of aquatic weeds in the Western Cape, in particular the city of Cape Town, but the systems down there are such that the winter rainfall tends to push them out, and this resets the system. And so it's a lot more dynamic. But what we have done in many of our systems, such as Hartbeespoort Dam and many of our other impoundments, is we've done these plants are adapted from the because of where they come from the Amazon basin, on big hydrological flow changes, and as a result of which they have, so many of them have these long-lived seeds. But what we've done is we've created stable flows. In other words, we put a large piece of concrete across a river. We've created these stable flows. So we've created that environment, and then we've just poured nutrients into it. And in the absence of any natural enemies, it really has been the trigger. That's not to say once we get on top of these, or once we get more on top of them, there are another list that are coming in. There's a newish weed on Hartbeespoort, Salvinia minima. We've got a biocontrol agent for that we've just finished testing. Egeria densa is a submerged weed. It's the first submerged weed that we've worked on. We've just about 3 or 4 years ago released a fly on that. We're watching how that's going. And Sagittaria, which is a wetland weed. We've just released a biocontrol agent on that. So there's plenty more in the wings. But as you say, some do well in some areas, but for these floating ones, you slow the flows, you increase the nutrients, and they're going to take off.
Okay. I don't see any more questions in the chat or any hands up. I'll give it a minute or two in case people have a question they'd like to ask. So, Professor Hill, thank you very, very much for your talk. It was, I found it absolutely fascinating. I'm a biochemist by trade, and this is somewhat outside my comfort zone, and I found it very interesting. Thank you very much for making it so accessible to a person like myself. I thank the members of the audience that have joined us for this Royal Society presentation this afternoon. I'd like to remind you that the talk is recorded and it will be available if there's aspects of it you want to see, or if you want to refer it to colleagues and friends and students, etc. It will appear on the Royal Society site in a few days' time. Please also remember that on the Royal Society site there are updates for all information regarding the Royal Society. I'd like to remind you that there are I'd like to advertise some upcoming talks in the next few months. Prof. Mike Bruton is going to be talking on the importance of Charles Darwin's visit to the Cape of Good Hope in May. Professor Gordon Brown from Medical Microbiology in Exeter is going to talk. Michelle Doherty from Space Physics at Imperial College. And then another person coming up is Professor John Compton from UCT Geological Sciences. So we have a nice range of talks coming in the future. So thank you very much for your participation today. Thank you very much, Professor Hill for a fascinating talk and for introducing me to a new form of measurement, the slip slop. And I look forward to reading more about your work in the press in the future. Thank you very much for people being here. If you're traveling somewhere, please travel safely. If not, I look forward to seeing you next time. Thank you and goodbye.
Thank you.